Thank you very much, Tracy, and hello to you all. I think firstly, I would like to thank the Australian Mycological Society, Tracy, but also Jonathan for providing a conduit for us to present some of our research to you all today. And I've just noted that this AMS presentation has also been live streamed into the 20th Mycology Colloquium. It's been a bit of an honor to present some of our work to you guys today. And I realized that that's going to encompass quite a broad audience, ranging from mycologists, and possibly we may have some serial plant pathologists dialing in as well as other molecular plant pathologists and traditional pathologists. So I certainly hope that the talk I present to you today is that I do it justice by trying to encompass as much of a broad interest group as much as possible. So the title of my talk is a little bit of an unusual one. It's called Untangling the Gordian Knot, right? And now, for those of you that do not know what a Gordian knot is, it is essentially a metaphor for a very difficult to solve problem that can be solved, or has been solved using a very, very unusual way to do it. And we relate this whole Gordian knot situation thing to some of the work that we did with a fungal pathogen of wheat, and how this fungal pathogen uses effector molecules to subvert the host wheat metabolism to its favor during the infection process, right? And so this whole knot, and no pun intended, will hopefully unravel throughout the course of the talk. Now I realize, and obviously I put this slide in a little bit last minute, knowing that we've also got the audience from New Zealand as well. So where are we located? Where are the Centre for Crop and Disease Management and me located? Where, unlike Tracy, who's located here, we are located on the other side of Australia, in the southwestern region over here, right? So this is, probably one of the most isolated region in the world, isolated capital in the world, and in Western Australia, apart from having plenty of iron ore and nickel as export products, we also have a acre crop such as wheat, barley, and canola, which is widely planted throughout the southwestern region of Western Australia, because we've got plenty of land. But I think in addition to that, we've also got quite a lot of fine wine as well in this region over here, right? So I think if you all are interested to pay us a little bit of a visit at Curtin University, feel free to get in touch with me. I'll show you around the place, and some crops, and perhaps some fine wine. But I think just get straight to the point. So this is a picture of the Australian grain belt. You can see here, this is where I'm located, and the whole grain belt extends all the way up to the eastern states as well. And we do have quite a lot of tonnage of wheat, barley, and canola being planted here. And the canola is probably the choice of crop at the moment, I think, simply because it fetches quite high price in the current market. But I think wheat still predominates as the #1 acre crop of choice throughout this region here, right? Now, whenever there's a monoculture, there's always going to be disease pressure. And one such disease that we've been, working on for the past, I would say, a couple of decades, is a disease called septoria, nodorum, blotch of wheat. Now, this disease over here, if you have a look at it, it's a result of lesions on the leaf, but also on the stem and bloom of the wheat as well. And these lesions over here, they coalesce, and what it does is that it reduces the photosynthetic capability of the crop, the wheat itself. And these lesions caused by this little nasty fungal pathogen called Parastagonospora nodorum, which we affectionately call it Stago, simply because we're 20 years working with this pathogen, you cannot pronounce its name properly. Now, this is a traditional necrotrophic fungal pathogen in that it's shoot first, ask question later, right? How it was that because in Western Australia we tend to practice a no-till policy in our cereal wheat field, And as a result, we do get quite a lot of stubble lying on the ground. And if these stubbles are infected by the pathogen in the previous years, when we start the growing season, what happens is that parathesia or pseudothesia gets generated on these stubbles, and then it produces these long distance dispersed ascospores, which gets blown around by the wind, and it lands on a newly germinated wheat plant, which when that happens there, it germinates and it starts forming these little brown lesions, which gets bigger and bigger and bigger throughout the growing season, and it becomes a problem. This is essentially the start of a problem, because during the growing season, when the lesion gets big, what the pathogen does is that it produces these asexual fruiting bodies called pycnidia, and these pycnidia produces pycnidiospores, which can then get splash dispersed by rainfall or raindrop, and these spores will land on green parts of the plant or another plant nearby, and the whole lesions and necrosis and stuff will continue to happen until harvest time. Okay? And I think because of that, it is actually quite a nasty pathogen that leads to quite significant yield loss if it's not controlled properly. Now, this is some of the economic figures that I pulled out from this study. Was published quite a number of years ago. Now, here, septoria nodorum blotch itself takes up a big pie of this economic loss in wheat per year. And this figure actually predominates in Western Australia, where septoria nodorum blotch is a predominant pathogen, and where I am at the moment, it is essentially a stronghold for the disease. The fungus tan spot, which is very similar to the nodorum, takes top spot. I won't talk too much about that today, but I think it's safe to say that our task is to actually try to reduce this slice of this pie to septoria nodorum blotch of wheat. Now, the good thing about nodorum is that your stag is that it is a very easy pathogen to work with, relatively speaking. Right? It grows rapidly. That it takes up pathogen in the space of a week and a half. It is also haploid. When it's haploid, it's actually got one copy of a gene. And if you want to knock the gene out or genetically alter it, that makes it easy because it's a single copy, right? We know that it can be you develop a genetic transformation protocol whereby we can put genes in or take genes out to knock out. We've also developed quite a lot of omic resources behind the pathogen as well to try to really figure out, on a global scale, how does the pathogen infect wheat, right? And one of these omic resources involve completion of the fungal genome, and this is a Herculean task which took a number of years to complete. And we started this whole process in 2005, where next generation sequencing wasn't really the hip thing back then, but instead we used a shotgun Sanger sequencing through the Broad Institute in Boston, whereby they've got a huge warehouse full of these capillary sequences, and it took a week to sequence the fungal genome through using this approach over here, right? So what we did 10 years later, we decided to finish off when next generation sequencing and proteomics and RNA-seq become, the technology, right? And as a result, in 2015, we basically stitched up some of the scaffolds from the initial genome sequencing, correct some of the base calling, and refine gene prediction. So following this, we then use technology and optical mapping to really get it down to the chromosomal level and complete the genome in around 2016. And we published a paper a couple of years ago on the finished genome. So you can see it's a really good resource to work with. Now a little bit more about the nodorum, the pathology of nodorum, and how does it actually cause these nasty lesions? How does it kill the host, right? So being a necrotroph, the pathogen kills the host tissue ahead of colonization. And this is really a typical cartoon of how a necrotroph fungal pathogen infect. So you have the spore propagule over here, right? It will germinate and it will try to get into the host itself through either directly through epidermal penetration, but most likely it will get in through stomatal entry. And once it gets inside the host itself here, you want to take hold of the epidermal and mesophyll layer by starting to produce these small molecular mass necrotrophic effectors, right? So these effectors itself, they will get into the host cell itself and will try to mess things up by altering the metabolism of these host cells in favor for the pathogen to infect. So we call this effector-triggered susceptibility, right? And as a result of these interactions, these host cells would essentially become necrotic and die. And because when it dies, it releases nutrients that allows the pathogen to start metabolizing and take into and allow to ramify itself through the host tissue, and therefore resulting in these nasty lesions that you've seen a little bit earlier on. Now, the pathogen system itself, the stagger wheat pathogen system itself, is partly defined by these necrotrophic effector host gene interaction, right? It's not hugely complicated to explain it, right? Let's just say if the pathogen itself over here possesses a particular necrotrophic effect or secretes it out, right? And these effectors are essentially small molecular mass, and they tend to have above average cysteine contents for stability purposes as well as folding, right? And the wheat itself requires the presence of a dominant susceptibility gene, and this could be receptor-like kinases and things like that. And if there's the presence of both these components, right, you get interaction. And if you do get interaction, you obviously get cell death and the manifestation of disease. If either one of these components, being the effector or the host gene component, are absent in the interactions, then obviously you don't get at the onset of the disease, right? Now, the pathogen over the years has been known to build up a huge repertoire of necrotrophic effectors through this side-by-side evolution with wheat itself, right? So here is a list of necrotrophic effectors. All of these are proteinaceous in nature, right? And these essentially interact with these particular wheat genes over here, right? The one in red has been cloned and mostly characterized, whereas the one that hasn't been cloned, the ones in black, it was still in the process of trying to clone them to try to see what they are, right? As you can see, I think the most immediate striking thing is that these effector receptor genes, they tend to interact on a gene for gene basis, with the exception of 267, whereby this one particular effector tends to interact with three different gene components in the wheat itself to elicit a disease response. Topsoil itself is a bit of a funny one. You interact with one of these particular gene in bread wheat, whereas this other one over here is a homeolog, which are found in the agelops, which is I think a tetraploid wheat, if I'm not I should know this if I'm not wrong, right? Now, because of the destructive nature of these effectors on wheat itself, we can actually exploit them against the pathogen, right? Because they've been cloned, we can put them into an organism such as E. coli and yeast and make synthetic version of these effectors, okay? And when we make synthetic version of these effectors, we tend to send out to breeders and pathologists alike to screen the wheat variety for reactions to these effectors, right? So you can see here, this is an example of a syringe filled with the effector inside here, right? And then we infiltrate it or pump it into the wheat leaf over here, leave it for a few days, and after a few days, you essentially get development of these typical necrotic symptoms. Okay, so remember, this is cell-free. This is just the protein only. There's no fungus in here. It's just the effector causing the necrosis, right? So this is what happened in a typical screening program, right? We sent it to breeders. They infiltrated it. They showed us this data over here. We recommend them to discard varieties that are sensitive to the effector and retain varieties that are insensitive to the effector, which possess a high potential for genetic resistance to the disease itself, right? So we call this effector-assisted breeding, right? And it's actually been a part for the past decade or so, right? Now, this begs the question, because we've got a huge number of effectors produced by the pathogen, right? So I think really the question is, do these necrotrophic effectors function quantitatively in a disease, right? And if you basically have a look on the other side of the coin, right? And if you basically remove these effective sensitivity gene in wheat, right, does that additively contribute to improvement in disease? In other words, do these effectors quantitatively work together, or are they functionally redundant, right? So we somewhat liken this whole effector tree susceptibility to this Voltron robot. If you're familiar with this 1980s cartoon series, and if you're not, right, it essentially entails a series of smaller robots that join into one big machine. And this machine over here is a much more powerful robot that goes around smashing everything in its path, right? So we try to understand if the effector works in a very similar fashion as this, which results in the in septoria nodorum blotch. And so what we did here was that we took a whole panel of commercial wheat varieties over here in Australia, in Western Australia, and these commercial wheat varieties, they are rated from very susceptible to moderately resistant, and these are the best ones we had, right? And what we did was that we infiltrate them with these three major effectors, right? The ones in red indicate strong necrosis, okay? Right? So these are, bad. I think immediately you can obviously see there's no correlation between the number of effector sensitivity and disease resistant rating. And I think to us, this is a little bit of a concern for us, because all along we've been advising breeders that, oh no, the effectors are your salvation for improving disease resistance, right? And yet over here, we don't really see a good, strong correlation, right? And we often get feedback from pathologists and other academics alike saying that, oh, we're not seeing TOX1, SN1 interactions out in the field, or TOX3, SN3, TOXA, TSN1. Do these effectors that you guys propose actually work in a field setting, right? So for a long time, we were wondering, why do we not see some of these interactions in the field? And we don't understand it. And I guess we essentially took quite a lot of these criticism and feedback on the chin to try to figure out, why is this the case over here, right? It turns out some of the studies that we perform in our lab, but also with a couple of collaborators in the States as well, through Tim Friesen and Justin Ferris from USDA, right? We actually figured out that the whole effector receptor interaction is probably a little bit more complicated in practice, in that it doesn't really function entirely in a quantitative fashion. But in fact, what these interactions do is that they actually inhibit the expression of other interactions in their contribution to the disease, right? For instance, TOX8 TSN1 has been shown to be antagonistic of TOX1 SN1, whereas TOX1 SN1 is antagonistic to the TOX3 SN3 and TOX2A SN2A, right? So this actually presents quite a little bit of a dilemma for us as to how on earth do we actually figure out which of these interactions are important in a field setting, in a disease under a field setting, right? Because when we advise breeders, we're going to have to tell them, right, which one of these should you guys prioritize in breeding out, because breeding for disease resistance in wheat, and in fact any traits in wheat, takes many years from start to end. It takes almost a decade for commercial releases, right? So we have to actually find a way to get our advice right to the breeders, right? And so at that time there, this whole knot theory was essentially laughing at us, and we had to actually find a way to try to untangle the knot, right? Now, there's a couple of hypotheses going about how these, what we call effector epistasis, right? Effector epistasis is where one of these interactions interacts with another interactions and antagonizes it, right? We hypothesize that some of these interactions may, in fact, feed into the common host signal transduction pathway that leads to necrosis. In other words, they're competing with each other, okay, to this one common pathway, okay? We haven't shown this yet, it's still hypothesis. What we actually have done recently is that we've actually done a little bit of work on this effector receptor interaction and this one as well, and shown their role in differential regulation between different pathogen strain that leads to this whole epistasis problem that you have here. And this is essentially an area which I'll spend the rest of my talk presenting to you all today, right? Now, a few years ago, we actually noticed that TOX1 expression varies significantly between different nodorum isolates, right? Our favorite Australian isolate, SN15, our model isolate, right? We actually noticed that This, along with another isolate from the US, produces much more TOX1 effector and two other cousin isolate, US isolate, SN4 and SN6, right? So this actually got us quite interested, right? Because we've got a reference genome, and what this actually allows us to do is to create or is to do a pangenomics on a number of the nodorum isolates, right? And with pangenomes, what this allows us to do is to align, to first look at the promoter sequence of the TOX1 gene as a first pass to look for variations in the promoter region that may explain for the differences in TOX1 expression, right? And so immediately when we align some of these isolates over here, we noted that there are minor genetic polymorphisms such as SNPs and small repeat, which at this stage here is irrelevant, right? But what we actually noticed was that this is this large indel, 401 base pair indel, right? And immediately we noticed it is absent in SN15 and SN4, SN2000, but present SN4, right? So this actually got us very much interested to see whether this is an element that dictates TOX1 expression between these two isolate variants over here, right? And we call this indel element P41, just for the ease of pronunciation. Now, before we go on and do some functional analysis, we had a look at the global distribution of P41. All right, we noted in the northern hemisphere, the nodorum that was collected in the northern hemisphere, right, it tend to possess P41, with the exception of some of the populations over here in America, right? In the southern hemisphere, much of the isolates collected tend to lack P41. And now I won't go into too much detail about this just yet, but what I'll do is I'll boomerang back towards the end and show this graph again and discuss about the relevance of the global distribution with some of the work which I'll be presenting over the next slide. So keep this in mind. I'll come back to it a little bit later, right? So now we start to do a little bit of functional analysis, right? So we took 5 strains, right? 2 of them lack P41 and 3 of them possesses P401, okay? The first thing we did was to look at the expression of TOX1 in these 5 strains under a in vitro condition that is conducive for TOX1 production. So immediately when we have a look at a gene expression, we noted that the strain that lacked P41 produces much more TOX1 than the strain that lacked these 3 strains that lacked P41, right? So one could argue that these 5 strains, the differences in TOX1 production in these 5 strains could be due to genetic variation in other parts of the genome, right? So we try to address that by creating isogenic promoter replacement mutant. And how we did this was that we created 3 different constructs. One of these constructs had P41, the second construct possesses P41, and the third construct also possesses P41, but the sequence in this region over here has been scrambled, okay? So we transform these 3 constructs and perform local-specific replacement of TOX1 in SN15 over here, but also in the wild-type strain 13072 as well over here as well, okay? And when we have a look at the TOX1 expression under the same condition, we noted that the isolate that lack P41 over here and here expresses TOX1 at a very high level. And again, when it introduces P401 here into these strains over here, we noted that TOX1 expression goes down. When we scramble the sequence, well, TOX1 expression goes back up. So this tells us that the repression of TOX1 expression is due to sequence specificity in this peculiar little element over here, right? So that was actually done under in vitro. So the next question is, what happened during infection? What happened during implanter, where it is actually where TOX1 expression is actually much more relevant, right? So we know that TOX1 is actually expressed much higher during the early stage of infection and goes down towards where the plant is a little bit, looking a little bit worse for wear. And this makes sense because you want the effector to be expressed during early infection, right? So we have looked at the expression of TOX1 with these 3 promoter isogenic mutants, right? And immediately again, we see isolate without P401 and with the scramble sequence here and here, it expresses TOX1 at a much higher level than the isolate with P41. And keep in mind, these y-axes are in log scale, so the fold difference on the linear scale is actually quite dramatic. Okay. next thing we did was that we actually do a little bit of work on the anatomy of the TOX1 promoter, right? So we did sequential promoter deletion to find out which area of the promoter are important for TOX1 expression, okay? And then we transform these individual constructs into our SN15 and directly replace the TOX1 locus, okay? It gets a little bit complicated to try to relate this to the expression of TOX1 below over here. So what I'll do is I'll summarize up for you, right? So when we did, promoter bashing, right, we actually noticed that this region over here isn't all that important for TOX1 expression, okay? This region over here where the Tata element is located, right, it is actually important for maximum TOX1 expression. Okay, so if you remove the Tata sequence over here, which makes sense, the TOX1 expression will go down dramatically, right? And what we have also shown is that this region over here appears to repress this region over here, right? So it's functioning again as a repressor region. We hypothesized that there's a transacting element that binds here and represses whatever that's happening over here, right? So I think enough of that for time being. So what we actually want to do now is to look at the importance of P41 under a real infection condition, which breeders and pathologists are concerned all alike, right? And so what we have with us at the moment is that we've actually got a wheat genetic population, double haploid wheat genetic population, right, which consisted of around 200 wheat lines that were generated from crosses that were made between these two parental varieties, Kalingari and Wild Casham. These two varieties are variety that has been used through commercial breeding purposes in the past, and they represent quite some of the original parental breeding stocks, right? Kalingari itself possesses TOX1 and TOX2A sensitivity, whereas Wild Casham possesses only TOX3 sensitivity. So if you cross these two and make the population, the gene that confers sensitivity to these three effectors will obviously segregate within the population. And this population was actually made by one of the breeding company Integrate, and it's been a godsend to us, right? So we genotype the population with genetic markers and use it a lot for genetic association mapping to look for particular traits in wheat itself that are favorable for breeding purposes, right? But in this case over here, it's disease resistance. So what we did is that we took these 200 lines and we infected with SN15 without P41, another word, high TOX1 expressing one, SN15 with P41, which is the low TOX1 expressing one, right? And we see what happened. So before we do any association mapping, we had a look at average disease susceptibility between in order lines, okay? That's infected by the high TOX1 expressing versus the low TOX1 expressing, okay? So you can almost immediately see the high TOX1 expressing isolate tend to be more pathogenic than the low TOX1 expressing isolate, okay? Which makes sense because the population got TOX1 sensitivity, right? And when we divided this population into varieties with TOX1 sensitive and TOX1 sensitive, you can immediately see those lines that possess all TOX1 sensitivity are much more sensitive to the higher TOX1 producer than the low TOX1 producer, right? And on lines that are insensitive to TOX1, the disease severity is more or less equal. So what ends up, right? High TOX1 expression drive SNN1 dependent disease, right? And the next thing we did was that we took all the data over here and performed association mapping, okay? So when we did association mapping of the wheat panel or the wheat population infected by the high TOX1 producer, right, we were able to detect through association mapping QTL where SN1 is, right? And this interaction actually contributes significantly to the phenotype of the disease, around 50%. So this is actually a very high disease contribution. Okay, so this makes a lot of sense, and we don't see other QTLs such as TOX3, SN2A, and stuff under this scenario. When we infect the panel with the low TOX1 producer, right, TOX1 SN1 contribute much less to the phenotype of the disease, whereas TOX2A now all of a sudden appear, right? We can now detect TOX2A SN2A interaction in its contribution to the disease at a low 12% contribution, although it's not as high if we can detect it, right? So what we actually have here is that when we alleviate TOX1 expression through the removal of through the inclusion of P41, we appears to somewhat alleviate the epistasis of TOX2A, SN2A in its contribution in septoria nodorum blotch. Another one, TOX2A, SN2A appears to function like a bench player if TOX1, SN1 is somewhat weakened by this P41 indel, right? And this make quite good sense for us on a plant breeding perspective. Now, this is our very simple working model so far, right? So you've got this low promoter region of TOX1. So you've got these two scenarios, right? In the presence of this P41, we hypothesize that there is a repressor element that actually binds here, which then represses or antagonizes a transcriptional activator that binds to the Tata region. And as a result, we have lower TOX1 expression, but in the absence of P41, we don't get the repressor to bind, and then the transcriptional activator will obviously have free reign to pump up TOX1 expression and drive up TOX1, SN1 dependent septoria nodorum blotch. Okay. Now that's not the end of the story just yet, right? So we were actually blessed because we live in the stronghold for septoria nodorum blotch, right? And because we live in the stronghold, we were able to get our hand on a temporal collection of the pathogen going all the way back to 1972, right? And much of the collection has been taken since post 2000. And what we're able to do is that we're able to monitor the distribution of PE41 within the population over time, right? And immediately you can notice that PE41 is maintained over here, in blue over here, at a very low level, relatively speaking, throughout this time over here, right? And because P41 result in low TOX1 expression and therefore theoretically less aggressive pathogen, right? Why is pathogen carrying this P41 being maintained throughout the time, right? Why has it not been replaced by these more aggressive isolates, right? So we set out to try to answer that particular question. There was a study that was carried out good 20-something years ago by Bruce McDonald, who's now in ETH Zurich, right? And what he did was that he did a population genetic study of the nodorum, around the world, but also within the same paddock as well. And what he actually found was that if he takes co-infecting the nodorum, right, or different lesions on the same plant, what he able to observe was that there are different nodorum genotype or different nodorum strain that co-infect the same plant, right? So the possibility, in my mind, was that if we have the same plant, we could essentially get isolates with and without PE41 infecting the same wheat variety out in the field. So we go out to test competitive fitness, right? So what we did was that we asked the question, does PE41 confer a fitness penalty in a mixed infection setting that we try to mimic in the field, right? And to do this, we used two strains, one without P41 and one with P41, high versus low TOX1 expression. Okay? We did single infection of each of these strains, but also a mixed infection on variety without SN1 and with SN1, TOX1 insensitive and TOX1 sensitive. Okay, we then did the infection and used digital droplet PCR to measure fungal biomass as an indicator of competition. So what we did was, when we have a look at a variety that's insensitive to TOX1 and have a look at the biomass, we noted that the biomass for these high and low TOX1 expressor, they are comparable. And when we did the mixed infection, they're also 50-50. Okay, there's really no advantage in the absence of SN1 or TOX1 insensitive. Okay, right? And the biomass of all these three conditions actually adds up to be the same, right? But this is actually where it gets a little bit peculiar, right? So what we have here is that we did single infection of the high TOX1 expressor versus low TOX1 expressor on a TOX1 sensitive wheat variety. And immediately you can see the high TOX1 expressor accumulate much more biomass than the low TOX1 expressor. But the surprising thing is that when we did a co-infection or mixed infection, we actually noted that the high expressor appears to keep the low expressor in the game. It is not eliminating or outcompeting the low TOX1 expressor in a co-infection setting, which is actually quite peculiar, quite odd for us as we thought. So I guess you can see so far that TOX1 function both of a selfish but also in an altruistic fashion. It is selfish is that it tries to compete against other effector receptor interactions, being epistatic to them, but it is also altruistic in that it keeps other TOX1 variant into the game, probably as a way to keep genetic diversity in the entire nodorum population in Western Australia, right? And this have quite a lot of consequences in how we advise pathologists and breeders alike in terms of controlling this disease at the genetic level. Now, going back to this world chart over here, right? And I guess all the work that we've actually done here somewhat starts to fall in line with what is actually observed in terms of the population structure, P41 prevalence, and also work that has been done previously on field diseases, right? So what does it really mean for TOX1, TOX2A, and field disease, okay? So what we did was that we have looked at some of the field studies that we summarized in this very nice paper over here by James Cochrane and Rowena Downey. Okay, so they summarizes field diseases where they use genetics to detect for the contribution of effector sensitivity in the importance in disease, right? So studies in Europe were only able to detect TOX2A, SN2A interactions out in the field. So this is a very important interaction out in field diseases in Europe, right? Whereas TOX1, SN1, not so much. You can find it here and there, but it is not an important interaction in field disease, right? Whereas studies that are carried out in Australia in the field setting were able to identify TOX1, SN1 interaction as well as TOX2A, SN2A. This observation actually lends itself quite well between the differences in P41 variation between these two subpopulations between Europe and Australia, right? I guess really, going back to the whole knot, we really untied the knot in general, right? The answer is no, we haven't. We probably have made a small little nick on it, but it's a good start, right? But there's also quite a lot of questions that still remains answered, such as how does TOX1, SN1 regulate TOX2A, SN2A at a mechanistic level? We still don't know that. We still don't really know, all this transactive element that interacts with the promoter region of TOX1. Also, what's the pathological explanation behind the global distribution of P41 as well? So we still trying to figure that one out, right? So what is the implication for our local ag community, right? And we recommend that over here we establish an annual fungal isolate collection program, disease monitoring, but also we need to monitor for changes in the frequency of P41 and effector gene as well in the local nodorum population, right? And we advocate to the breeders and pathologists alike to remove SN1 and SN2A sensitivity gene in their wheat germplasm as well to hopefully improve nodorum resistance. But also stacking 4 independent and relevant resistance allele provides the most optimal resistance to septoria nodorum blotch. And this is according to some of our study and collaborators as well. And we also recommend using relevant and new nodorum isolate to test for cultivar resistance before commercial releases as well, right? So I guess that's all from me, and it's safe to say that much of the work that I presented to you today has been carried out by our ex-PhD student, Evan, who's just left our lab for greener pasture not too long ago. But obviously the work is not made possible without funding from the Grains Research Development Corporation and Curtin Uni, right? But also we thank collaborators from CSIRO, University of Neuchâtel, Max Planck, and also Integrate for providing that superb magical population that help us answer everything that we need to answer. And I guess thank you all for your attention, and I'm more than happy to take any questions that you may have.
Thanks so much, Casey. That was really interesting. A bit of a bias there since I'm working with Parastagonospora fungus too at the moment. So thank you for that insight. I'm going to kick off the questions. So if anyone has something that they want to ask, please go ahead and input it in the Q&A function or raise your hand and I'll get to you directly. To start off, what do you suspect is the difference between the northern and southern hemisphere effectors you found? Do you think it's something to do with climate or the preferred breeding lines? What's your suspicion?
Oh, I mean, there could be many things over there. Look, I think one thing that we haven't really looked at is the the prevalence of SN1 in the commercial varieties in the northern hemisphere versus the southern hemisphere over here. So the presence of SN1 could essentially be acting as a selection pressure for that drives the proliferation of loss of P41 within the population. So that's something that we haven't really looked into in details yet, and you know, at the end of the day, that's just the one hypothesis. You know, apart from that, we really have no idea. We're not really, you know, population genetic expert per se, but one of our collaborator from Switzerland is, and I think clearly he's. We had a good long chat with him, and I think still he's working in this area for a very long time. He still don't have any idea at this stage here. But I think the first thing first is to essentially look at the prevalence of SN1 in wheat that are widely planted between these different regions to see if they are really the ones that act on TOTS1.
Yeah, I'm assuming that the countries in the northern hemisphere have prefer different breeding lines anyway. So there's got to be some kind of genetic difference, surely.
Yeah, there would be. Yeah.
Yeah. Okay. No, very interesting. Anyone coming through with questions? As I said, you're welcome to raise your hand there. I know that there's a full schedule ahead for those of you who are part of the mycology colloquium. So you're probably going to be off to lunch, or is it afternoon tea there pretty soon? But yeah, I think that's it looks like that's it for now. Hang on a second, we've got someone up. Just David Orlex basically saying thanks for an awesome talk. And thank you to David for having us as part of your colloquium. We really enjoy cooperating like this. And as you may have seen at the start of the talk, we had a bit of a slide advertising the conference we're going to be holding at the end of the year, and that's with the New Zealand Microbiological Society as well. So please look up that online. You can join us. I think that's it. So thank you so much, Casey, for joining us and for presenting your research. Feel free to get in touch with Casey if you have any further questions. And if you'd like to present your research in one of our seminars, please get in touch with me via the Australasian Mycological Society website. Thank you very much.
Okay, no worries. Thank you very much. Okay, see you later.
Bye.